Hello, class, and good evening from my side. Okay, so we have to continue talking about microorganisms, and so as you remember, we still have to finish talking about the viruses. How a very convenient time. Then continue talking about bacteria and then about those eukaryotes, which are parasites such as fungi, protozoan, and those helminths. There are no algae, which are parasites. Some algae can parasite on fish, but I will leave you slides of algae, but just so you know, there are no non-parasitic algae on humans. Um, so just a short reminder, remember we said that viruses are obligate intracellular parasites, which mean they need to invade a living cell in order to replicate. I mentioned that they possess those H and N spikes on the surfaces. And then that they can be either DNA or RNA viruses. Inside the capsid, you will see that the capsid is made out of capsomeres, so these small protein subunits, and then inside there is either DNA or an RNA. And usually, because they're so small, they range in nanometers. They might have several enzymes, but usually there is nothing in there. So since they don't have cytoplasm, no organelles, no nothing, that is why they need to invade a host cell and then hijack the system to replicate their nucleic acid and then biosynthesize all the subunits. We are not it's very hard to grow them in a lab. Remember, I was telling you that you have those empty spaces where the bacteriophage killed off the bacteria. So bacteriophage is a special virus that is attacking only bacteria. So you have DNA and RNA viruses, which can be naked and enveloped. This phospholipid bilayer doesn't come from them because they cannot make it by themselves, but they stole that from their host that they invaded. So we are going to continue in details from here. So the usual testing for viruses is something that we mentioned. It's called a PCR, a principal component analysis, where you multiply little pieces of DNA, and that's how you compare them to the known DNA strands. So, just because we're in this situation right now, there wasn't a PCR machine in the Bahamas, but now there is one. So now Bahamas are equipped with a PCR machine, and we are able to detect the coronavirus. In the absence of the PCR machine, what we are really looking at are those changes that viruses cause to cells, so-called cytopathic effect. So usually a cell that is invaded with a virus, you can see some sort of unusual granules around the nucleus called inclusion bodies. So this image right here, that's a chicken embryo, and then you can see how nucleus is kind of it's deformed and has this unusual spots. So these indicates the presence of a virus. And also inside here, which is a cross section of some tissue, not sure exactly which one, but here again you can see, so this is supposed to be a nucleus, and this right here, it's a nucleolus. So you see all these small little purple dots, which are inclusion bodies. So that is how you detect the presence of the virus without using the PCR machine. So those spikes that I mentioned, H and N, so you have hemagglutinin spikes and neuraminidase spike, and then inside there you have a DNA or an RNA. This is an example of AIDS virus. So right here, I am going to try to end this show and then tell you to go to Dropbox. Oh, excuse me. Sorry about that. Go to Dropbox, where I posted an extremely good video on how AIDS virus is replicating. So AIDS, HIV. It's a retrovirus, so it's an RNA virus. And here you can see a very detailed animation on how does a retrovirus replicate. Okay, so right here is when you were supposed to watch this video, please. I already posted that on your Dropbox, so right here it should be there. Okay. And right here, this is an interesting story about a virus which was named H7N9, which was a bird flu back in 2013, so not such a long time ago. So what you can see here is there were several different types of viruses which were attacking domestic animals and wild animals. And then this virus mutated in a sense that it obtained a genetic material from all of those hosts. And now this virus contained the genetic material from the previous host, and now it mutated enough so that it can start attacking humans. For the coronavirus, we are unsure exactly where did it originated from. I mean, which animal. It was a zoonosis, but they're still not sure which animal, because the virus, by now there are already 2 strains of the coronavirus. So when they compare the genetic structure to known hosts, there was no exact matching. So they have 2 suspects. So it's either bats or snakes. Chinese people, they eat both of them. So they ain't sure whether the virus originated from a bat or a snake. But again, like I said, the coronavirus mutated and then went from wild animals onto the humans. So this is a story that's been repeating a couple of times, and then you will see that you have specialized viruses that attack, horses, cattle, pigs, whales, ducks, cats, sea lions, and so forth. And as you can notice, they're all named based on their H and N spikes. This is why influenza virus changes its genetic structure every year as it jumps from human to human. That is called antigenetic shift. So, and that is one of the reasons you need to be vaccinated for that particular strain of the virus each year. In the next video, I'm trying to keep videos short so that I can upload them to YouTube. We will be talking about the viral replication and what is their cycle.